Assalamualaikum and selamat sejahtera. My name is Nur Akrimah binti Hasbullah. My matrix number is PC09-2053. Today, me and my friend will talk about surgical gown sterilization. For the most suitable method for surgical gown sterilization is moist heat. And my friend will explain why the reason we choose the method for surgical gown sterilization is moist heat.
Hi, my name is Putri Nurasha binti Zainuddin, PC 09200089. Today, I want to talk about surgical gown sterilization. The reason of my choice is moist heat method are suitable to sterile surgical gown because they have bacteria and viruses are made up of proteins, and moist heat sterilization works by denaturing these proteins from surgical gown.
I will explain the mechanism of action of the method. Moist heat destroys microorganisms by the irreversible degeneration of enzymes and structural proteins. The temperature at which degeneration occurs varies irreversibly with the amount of water present. Sterilization in sterilized steam thus requires precise control of time, temperature, and pressure. First step is water is added on the bottom of the autoclave and the article to be sterile are placed in a perforated shelf. Second, the lid is closed and locked. Third, a vacuum pump sucks the air from the chamber and the air from the inside autoclave has been removed. 4. Steam pressure rises inside and the temperature reaches the correct level. 5. Steam kills bacteria and microbes. 6. When the holding time is over, the heating is stopped and autoclave is allowed to cool till pressure goes indicates. 7. Steam is released, the door is opened, and air is allowed to remove from the autoclave. 8. The lid is opened and the sterilized articles are removed. 9. Items are ready for use.
The apparatus or device that used in this method is autoclave at 134 degrees Celsius. The advantages of moist heat is it is more effective to kill the microorganism and has better penetrating power than dry heat, and it rapidly heats and penetrates fabric and also have short procedure time. Okay, that's all from me. Thank you.
For the disadvantage, moist heat autoclave is 1. The moist heat will moist retention. For the second, carbon steel can get damaged due to the moisture exposure. 3. Only stainless steel instrument and plastic which can bear the heat be sterilized. 4. The load is damaged by autoclave. That's all for me. Thank you.
Assalamualaikum, my name is Nur Najwa binti Sana. The matrix number is PC09200077. A vaccine is a preparation that is used to stimulate the body's immune response against disease. Irradiation was essential in vaccine production. Radiation sterilization has been used to develop a variety of vaccines because it can eradicate chemical contaminants and penetrate pathogen to destroy nucleic acids without damaging the pathogen surface antigen. To produce vaccines of high quality, we need to perform sterilization during the production, which is done through a form of irradiation. High dose of gamma rays can be used for the development of vaccines that exploit inactivated viruses. Gamma irradiation sterilization machine. Sterilization with gamma rays is one of the most reliable, effective, and common alternative sterilization methods used today. Measuring the quantity of vaccines can increase the chance of recovery of COVID-19 patients. Therefore, irradiation is an important element in defeating the virus. The different doses of gamma irradiation vaccines can cause different impact to human body. That's all from me. Thank you.
Assalamualaikum. My name is Nurul Huda binti Matari and my registration number is PC09-2084. Pasteurization is the process of heating something quickly, then cooling it again. Pasteurization means destroying 99.9% of disease-causing mechanism and extends shelf life to 60 until 21 days from the time easy packaging. Pasteurization increases the storage life of milk because it reduces lactic acid production microorganisms. Enzyme milk are destroyed by pasteurization, and that is how milk can be stored safely for a long time. Pasteurization kills pathogens and reduces the number of spoiler-causing microbes while maintaining food quality. HTST presentation equipment use sanitary plate heat exchanger utilizing hot water or steam to raise the temperature of product. The presentation temperature and the time depends on the products. Advantage of presentation is kill pathogen, decreases storage period, and deactivates enzyme. The advantage presentation is don't doesn't kill heat-resistant pathogen and reduction in the nutrition contents. That's all for me.
Hi, Assalamualaikum. My name is Nurifa Ilianis binti Sahrul Rizam. My metric number PC09200074. The most effective method of sterilization of prefilled syringes today is predominantly accomplished using gamma radiation. While these methods are well established, their limitation could impede innovation and progress in the pharmaceutical and biotechnology industries. The reasons are radiation kills germs that can cause disease and neutralize other harmful organisms. The mechanism of action for gamma irradiation is a physical or chemical means of sterilization because it kills bacteria by breaking down bacteria DNA, inhibiting bacterial division. Energy of gamma rays passes through the equipment, disrupting the pathogen that can cause contamination. Next, the apparatus of gamma irradiation sterilization is performed by exposing the product to a radiation source, typically cobalt-60 isotope, which decomposes into nickel-60 isotope, firing of gamma rays in the process. Okay, advantage for gamma irradiation are does not leave a toxic residue in the products, final packaged products, and no significant increase in temperature. Okay, so for disadvantage of gamma irradiation are in small quantity, more expensive, but at large quantity and comparable place with other sterilization mode.
Assalamualaikum and hi. My name is Nurul Hazliahani binti Ahmad Aswazir and my metric number is PC09200082. So now, I will explain about the sterilization of powder. Firstly, the most suitable method for sterilization of powder is dry heat sterilization. It is because dry heat sterilization uses high temperatures to kill microorganisms and bacterial spores. Dry heat sterilization requires higher temperatures and a longer application to accomplish the same result. Next is the mechanism of action of dry heat sterilization. The mechanism of action is kills the organisms by protein denaturation, oxidative damage, and also toxic effects of elevated levels of electrolytes. There are several different types of dry heat sterilization, such as hot air oven, incineration, and also flaming. This method also has its advantages and disadvantages. This is the advantages of dry heat sterilization. First, it penetrates the materials. Second, it is non-toxic and does not harm the environment. Third, it has no corrosive effects on instruments. And the last one, it has relatively low operating costs. Disadvantages of dry heat sterilization. First, it may damage some items due to high temperature. Second, it is not suitable for surgical dressings. And the last one, it has long sterilization period. That's all from me. Thank you.